This is the fallopian tube of the female reproductive organ. The fallopian tube contains three parts: the funnel shaped infundibulum, the slightly swollen ampulla, and the part that connects the fallopian tube with the uterus called isthmus. It is the ampulla of the fallopian tube where the process of fertilization takes place. However, what exactly is fertilization? In simple terms, fertilization is the fusion of male gamete with female gamete. The pronucleus of the male gamete fuses with the pronucleus of female gamete to form a diploid zygote. So, for the process of fertilization, what do we require? We require two things. Number one, the male gametes, and number two, the female gamete. So, let's look at both of them one by one. First, the male gamete. During one ejaculation, and I repeat, one ejaculation, around 200 to 500 million sperms are ejaculated. However, from these many sperms, that is around 200 million sperms, only and truly only around 200 to 500 of them reach the ampulla. So at the region of ampulla, out of 200 million sperms, only 200 reach. Only so few of them reach the ampulla. Second, so during ovulation, the secondary oocyte gets ovulated out. There are these finger-like structures that are present at the end of infundibulum, and these finger-like structures are called fimbriae. These fimbriae, they show sweeping movement, and the sweeping movement of the fimbriae helps the fimbriae to collect the secondary oocyte from ovary. So this is what happens. At the time of ovulation, a bump is created at the site where ovulation is about to occur, or at the site from where secondary oocyte is about to come out. At this particular site where there's a bump, the fimbriae starts to sweep and sweep and sweep on the surface of that particular site, so that as soon as the secondary oocyte gets ovulated, the fimbriae is able to collect the secondary oocyte and send it inside the infundibulum. So now we know the collection of the male gamete right here and the female gamete right here. However, once the gametes have been collected, they need to reach the ampulla for the process of fertilization. The sperms that have been deposited need to travel through the cervix and then the uterus and then finally reach the fallopian tube. The secondary oocyte needs to travel through the infundibulum and reach the ampulla. And if you can compare both of them, you can see that the sperms relatively have to travel a longer distance as compared to the secondary oocyte. So let's look at the movement of both of these gametes. First, the movement or the travel of sperm. The movement of sperm. So there's a misconception. The misconception being sperm travels through the uterus majorly by the proportion of its tail or the flagellum. However, that's completely wrong. Yes, the tail helps in the propulsion of the sperm, that's right, but that's not the main reason. The main reason for the propulsion of the sperm is the peristalsis of uterus. The wall of the uterus contains smooth muscles. The smooth muscles, they contract and relax, creating a peristaltic movement. This peristaltic movement helps the sperm migrate through the uterus and reach the fallopian tube. And very little by the propulsion of its tail called flagellum. Propulsion by flagellum. Once the sperm reach the start of the fallopian tube, they inactivate. They become inactive. They deactivate. Second, let's look at the movement of the secondary oocyte. Once the secondary oocyte is ovulated out, it is collected by the sweeping movement of the fimbriae and it enters the infundibulum. Then what happens? Movement of secondary oocyte. So the main reason for the movement of secondary oocyte is the peristalsis of fallopian tube. So the wall of the fallopian tube contracts and relaxes, which helps the secondary oocyte to propel forward. And the movement is also helped by ciliated columnar epithelium, which lines the internally the fallopian tube. So the wall of the fallopian tube is internally lined by these ciliated columnar epithelium, and the cilia of the columnar epithelium show rowing movement, that is to and fro movement, like the rows of a boat. And this rowing movement helps to migrate the secondary oocyte through the infundibulum. Okay. So now the secondary oocyte has reached the ampulla, and so has the sperm. And so the fertilization has to occur. However, the fertilization does not occur yet because the sperm is still stuck here. It hasn't reached here. Just because it's in the diagram, that doesn't mean I have reached that point of explanation. See, the sperms are still inactivated and they are deactivated at the region of isthmus. These inactive sperm becomes active. How? So a certain part of the secondary oocyte called cumulus ooferus secretes chemoattractants. Chemo refers to chemical, and attractants cause it brings about attraction. The chemoattractants released by the secondary oocyte attracts the sperm and activates it. As a result, the sperms become motile once again and they reach the ampulla. Okay. Now finally the egg or the secondary oocyte and the sperm, they have reached the ampulla. At the ampulla, the sperm can't fertilize the egg yet because it is not ready physically. It goes through two different processes so that it can finally fertilize the egg, and they are number one, capacitation, and number two, acrosomal reaction. Let's look at both of them one by one. For the sperm to fertilize with the egg, it has to go through two different changes. First is capacitation. Once the sperm goes through capacitation, it can finally go through the second process, which is called acrosomal reaction. First, let's look at capacitation. So capacitation refers to the process where the sperm finally gains the capacity to penetrate the outermost layer of secondary oocyte, which is corona radiata. This crown-like surface or crown-like layer surrounding the secondary oocyte radiating outwards is corona radiata. Sperm by itself can't penetrate through this corona radiata without first going through the process of capacitation. During capacitation, the sperm goes through a 7-hour conditioning. 7-hour conditioning, so that the sperm is finally ready to penetrate through corona radiata. And this 7-hour conditioning of the sperm is what we call capacitation. So what actually happens during capacitation? See, this right here is the head of the sperm. The head of the sperm contains nucleus. On top of the nucleus, there's a cap-like structure called acrosomal cap. The acrosomal cap contains the acrosome, which is covered by the acrosomal membrane. And then finally, there's the plasma membrane. There's one more extra layer, which is made up of glycoprotein, which we call the glycoprotein layer. Due to the presence of this glycoprotein layer, the sperm initially is unable to penetrate through corona radiata. However, after going through the process of capacitation, the sperm loses its glycoprotein layer and finally gains the capacity to penetrate through corona radiata. After the sperm penetrates through corona radiata, the head of the sperm attaches itself with a protein receptor that is present on the surface of zona pellucida. So this layer right here is what we call zona pellucida, which is present right below corona radiata. The surface of the zona pellucida contains these protein receptors. To these protein receptors, the sperm gets attached, or the head of the sperm gets attached. And the name of this protein receptor is GP3 protein receptor. That sperm binds with zona pellucida. And number two, after the sperm finally gets connected with the ZP3 protein of zona pellucida, it can undergo acrosomal reaction. So until or unless the head of the sperm attaches with the zona pellucida protein, it can't undergo acrosomal reaction. For the sperm to undergo acrosomal reaction, it has to first get attached with the GP3 protein. So after capacitation comes acrosomal reaction. So the capacitated sperm, which has finally penetrated through corona radiata, goes through acrosome reaction so that it can penetrate through zona pellucida. What happens during acrosome reaction? So here's the head of the sperm, which contains nucleus and the acrosomal cap, which contains the acrosomal membrane. Attachment points are created both on the plasma membrane and the acrosomal membrane. At these attachment points, the two membranes fuse. As a result, the head of the sperm, this area gets removed due to the formation of pores, and the sperm finally looks something like this. Inside the acrosomal bag or the acrosome, there are enzymes that are present. Two major enzymes, acrosine enzyme and hyaluronidase enzyme. These two enzymes help the sperm to finally penetrate through zona pellucida. So here you can see in the diagram, here's your secondary oocyte. Again, the female gamete is still stuck in secondary oocyte. It has not gone or undergone the process of meiosis 2. It is still stuck in the metaphase 2 of meiosis 2. So here's the secondary oocyte, and it is surrounded by zona pellucida. The sperm has penetrated through corona radiata after going through the process of capacitation and the sperm gets attached to the GP3 protein. After the sperm gets attached to GP3 protein, it also undergoes acrosome reaction, during which attachment points are created both on plasma membrane and the acrosome membrane, where the two membranes they attach with each other, forming various pores. And it is through these pores that enzymes acrosine and hyaluronidase are secreted out. With the help of these two enzymes, the sperm is finally able to penetrate through zona pellucida. The head penetrates through zona pellucida. So what happens after the sperm penetrates through zona pellucida? So first, the sperm, let me write the steps as well. So sperm penetrates zona pellucida. After the sperm penetrates zona pellucida, sperm touches the oocyte membrane, the secondary oocyte membrane. So here's the secondary oocyte, which is surrounded by zona pellucida. The sperm finally penetrates through zona pellucida. As soon as the head of the sperm touches the surface of the secondary oocyte, the secondary oocyte immediately completes the process of meiosis 2 and forms the egg. Then something called cortical granules, they release lysosome. As you can see in this diagram, at the cortex of the female gamete, there are these granules that are present, and these granules, they are filled with lysosome enzyme. As soon as the head of the sperm touches the membrane of the secondary oocyte, two things happens. First, the secondary oocyte completes its second meiosis division to form egg, and secondly, these cortical granules they secrete lysosome. What does lysosome do? These lysosome they, let me write it here. So these lysosome they deactivate deactivate the GP3 proteins, as a result of which no other sperms are now able to penetrate the egg. Only one, and I repeat, only one sperm is able to penetrate the egg. So there's no tension of polyspermy. Poly means multiple and spermy refers to sperm. So multiple sperms can't fuse one single egg due to the deactivation of GP3 protein by lysosome secreted by cortical granules once the sperm touches the secondary oocyte membrane. So what are the two things that happens as soon as the sperm touches the secondary oocyte membrane? First, the secondary oocyte immediately completes meiosis II division to form egg. Second, the cortical granules of the egg secretes what we call lysosome, and these lysosome they deactivate the GP3 protein present on the surface of zona pellucida so that no other sperm are able to attach with it or penetrate the egg. After that, no more sperm can penetrate. See, when the sperm penetrates the egg, only the content present inside the plasma membrane, the cytoplasm, organelles, and all that stuff enters the egg. The plasma membrane is left behind. In case of human, even the tail gets penetrated inside the egg. However, the tail later disappears. What remains is only the head region, and the head region contains the male pronucleus, while the egg contains the female pronucleus. The female pronucleus and the male pronucleus, both of them are haploid. The haploid female pronucleus and the haploid male pronucleus fuses to form a diploid zygote. And this is how we complete the process of fertilization. Let me revise the whole thing for you in very short. Okay, so fertilization takes place in the ampulla of the fallopian tube of female reproductive organ. Around 200 million sperms are deposited from one ejaculation. The sperms reach the fallopian tube with the help of the peristaltic contraction of the uterine wall, and by very little with the help of the propulsion by its tail. Once the sperms reach the beginning of fallopian tube, they become inactive. After ovulation, the secondary oocyte gets ovulated out, which gets collected by the sweeping movement of fimbriae. Once the egg reaches inside the infundibulum, it travels with the help of the peristaltic movement of fallopian tube and the cilia of the ciliated epithelium and reaches the ampulla. The certain part of the secondary oocyte called cumulus oovarus secretes chemoattractants that attract the sperms and activate the sperms. The sperm finally reach the ampulla as well, and they are ready for fertilization. However, the sperm is yet not prepared enough to go through the process of fertilization. It has to go through two changes. First, capacitation, and second, acrosomal reaction. During capacitation, the sperm gains the capacity to penetrate through corona radiata. Corona radiata is the crown-like outermost layer of the secondary oocyte radiating outwards. For the sperm to penetrate through corona radiata, it has to go through a 7-hour conditioning process which we call capacitation, where it loses the outermost glycoprotein layer. After the sperm penetrates through corona radiata, the head of the sperm binds with a surface protein present on the surface of zona pellucida called GP3 protein. GP3 protein are responsible for two things: for the sperm to bind with zona pellucida and for the sperm to undergo acrosomal reaction. As soon as the sperm binds with GP3 protein, it starts acrosomal reaction. In acrosomal reaction, attachment points are created on plasma membrane as well as acrosome membrane, which attach with one another, forming pores. From these pores, acrosin and hyaluronidase enzymes present inside the acrosome are released out. With the help of these enzymes, the sperm is finally able to penetrate through zona pellucida. As soon as the head of the sperm touches the surface of the secondary oocyte, the secondary oocyte immediately completes meiosis II division to form egg. As well as there are granules called cortical granules present at the cortex of the egg that release lysosome. The lysosome deactivate the zona pellucida proteins or GP3 proteins. Once the GP3 proteins get deactivated, no other sperms can now attach with zona pellucida or penetrate the egg, so that polyspermy does not occur. During penetration, only the internal content of the sperm gets inside the egg, that is the cytoplasm and the organelles. Both the head and the tail enters inside the egg. However, the tail region later degenerates. The head region contains the male pronucleus and the egg contains the female pronucleus. Both of them are haploid. The male pronucleus and the female pronucleus fuse with one another to finally form diploid zygote. And this completes the process of fertilization.